Hi, everyone. Welcome to the ASF Science Podcast. Thanks for joining us. There are a couple great studies to report this Friday. One that looks at brain function as it changes with intervention, and the other which looks at the brains of people with autism as they get older. They're informative to each other, believe it or not, and hopefully I can explain why more at the end of the podcast. The first study is led by Pamela Ventola at the Pelfrey Lab at Yale. Her goal was to better understand how differences in brain function may lead to a differential response to intervention in individuals with autism. So we all know that autism is different in everyone, and it's no surprise that the brains of people with autism vary too. She used a technology called functional magnetic resonance imaging, or fMRI, to study brain activity in different regions. They found at the start of an intervention study, some people with autism had higher than expected levels of brain activity in one area called the posterior temporal sulcus. The other had lower than expected levels of activity in that same area. So this was prior to receiving any sort of intervention. This posterior temporal sulcus, if you point on your head, it runs from right about behind your ear to the back of your head. So this finding wasn't expected or premeditated. It just happened that there were two different groups of kids that had exactly the different profiles in the same area of the brain. So despite these differences, they all had an autism diagnosis, which is pretty amazing in itself. After they did all these baseline differences, they gave everyone 4 months of what's called pivotal response therapy, or PRT. This is a behavioral therapy which is based in ABA and has previously shown to improve some of the core symptoms of autism. It's play-based and targets critical or quote-unquote pivotal areas like motivation, initiation of social interactions, and self-management. The approach plays on what motivates each child and builds on that in a naturalistic play setting. After 4 months, they put them back under the fMRI scanner again. There were two incredibly important findings. First, they found that PRT improved functioning in everybody. Everyone in the study showed improvement. All of the improvements weren't statistically significant, but everyone showed some sort of positive change. They looked at social, communication, and adaptive skills. So this is more evidence that PRT is an effective treatment for autism. The second important finding was that brain activity was changed in both groups. It turned the activities levels more towards what you would normally see in kids without autism. So here's a behavioral therapy that works and is mirrored by brain changes. So for example, those individuals had quote-unquote hyperactive or higher than expected levels of activity in that brain region. It dampened down or turned down that response. For the individuals that had less than expected levels of activity, or the hypoactive group, it turned up the response. In scientific terms, this is called normalization. I know that that actual term isn't popular with everyone, but that's what it's called. There was some variability in the way each child responded to PRT, and even though it didn't work the same in everyone, you wouldn't expect it to, and it did work. The important thing is that using brain imaging may be able to identify high and low responders to other types of therapy in the future, including drug therapies. If you want to hear more about the study, the lead author, Pam Ventola, who's ASF funded, wrote a blog post for the ASF website and actually included pictures of her delivering the therapy. So please go to the ASF website and check it out. The second study comes from a group that pooled all of their brain imaging data across different sites and different time points to look at change in brain structure over age. It was led by Nick Lang at Harvard, but the data came from a lot of other places. It was a large collaborative study. They combined data from a longitudinal study where people's brain sizes were measured, and then these individuals came back twice again over the course of 8 years. They were then compared to a group of people without autism. Everybody was around 3 to 35 at the start of the study, so they tracked them until they were about 11 to 43 years of age. I have to say I was a little disappointed that they only reported male data. There weren't enough females to properly compare them to, and so we'll actually have to wait until enough are recruited. So unfortunately, there aren't any sex differences to look at here, but hopefully we'll be able to look at them in the future. So I mentioned the word longitudinal, so I'll explain. There are two ways actually you can measure change across time. First, you could get completely different groups and have them come in at different time points. So one group comes in at the first time point, and a second group comes in at the second time point, and the third one comes in at the third time point, et cetera, et cetera. But actually, the more interesting way to look at it is to take the same person and look at their changes over and over again. This allows for actually a more accurate calculation of what brain regions are changing and when, and can compare it to people without autism and with autism. In this study, a lot of people stayed for all 3 scans, which is pretty incredible. But as you'd expect, some of them only came in for 2 scans, and a small group only came in for 1 scan. So the main findings. With age, as you all probably know, the brain volume of many regions declines. That's no real news here. Your brain volume is highest when you're about 10 years of old, but once you get to be about 10, you start the slow decline in brain volume. I know, that's pretty depressing. When it really starts to decline depends on the actual brain region. In some regions, it starts to go fast in the 20s. In others, it's the 30s and the 40s. But what happens if you're a man with autism? The decline starts earlier. The 2 groups, autism and typically developing, show a clear separation of brain size by age 30. That means by 30 years of age, the size or the volume of the brain is reduced in brains of people with autism compared to those without autism. So one thing you may be saying is, haven't I read that people with autism have big brains at birth? So this early decline may actually make sense. In this study, this was the case in only about half the individuals with autism. They started off bigger and then kind of got into quote-unquote typical values between 10 to 15 years of age. But that's when you started to see that abnormal or atypical further decline into adulthood. So this is all interesting, but what's happening to the cells? Well, there's a different study, not this one, but one that was published about a month ago, that looked at cell size and brain regions across ages. These data came from individuals with autism who died, and then their families decided to donate their brains for autism research. This is an incredibly precious and rare resource, but it's really the only way to answer these questions. Jerzy Van Gel at the Institute of Basic Research in New York took brain samples from individuals with autism from ages 40 to 60 years of age. Again, this is not the same people as in the first study, and the sample size was much smaller. We're talking hundreds in the Lang study and maybe 10 to 15 in this study. What they found that as people with autism age, the size of their cells get bigger. So taking the data from the Lang study with this study, you can see smaller brain regions in adults with autism with actually bigger cells. This smaller region with bigger cells packed in there might explain the worsening of symptoms of some people with autism over time. It oversimplifies what's really going on, I know, but it certainly reaffirms that autism is dynamic and changing. I wanted to present you with both of these studies together because the Lang study showed a decrease in brain structure over time compared to typically developing individuals. But the Ventola study showed that despite changes in size and function, the brain is plastic and very receptive to intervention. We really need to better understand the role of environmental enrichment intervention on brain activity, both on an activity level and on a cellular level. Because the brain is responsive to intervention in people with autism, that may actually change the trajectory of adolescents and adults with autism. Thank you for listening this week. Please stay tuned for updates, and please go to the Autism Science Foundation website to read Dr. Ventola's blog. It's a really great one, and I promise you she does a much better job explaining the study than I did. Have a great weekend and talk to you next Friday.